Ron, am I audible? So good afternoon, everyone. So on behalf of Indian Society of Clinical Research ISCR, we are going today to celebrate Maharshi Charak Jayanti Mahotsav. So with collaboration of Indian Pharmacological Society West Bengal Chapter and iNovocare E-Academy, we are organizing this today. And we are really honored to have a galaxy of faculties with us today. And just 3 days before Independence, 75th Independence Day, we are really proud to host this one of the most important treasure of India, that is Ayurveda, in front of us. So we all know the very important sloka from our long days. We were taught that Sarve Bhavantu Sukhina, Sarve Santu Niramaya, Sarvo Bhadrani Pashvantu, Ma Kashit Dukha Bhagavat. So we have a tradition to help all. And for that, one of the most important treasure we have is Ayurveda, our long history with medicine. And for that, Maharshi Charaka was the father figure. So we are celebrating today his Janma Jayanti. So may I now request our President of Indian Society of Clinical Research, Dr. Sanis Davis, to have the welcome address for all of us. Welcome, sir.
Hi everyone, hi. Sanish, we can't hear you. This is Dr. Subhrojyoti Bhowmick. And I'll request Shambo to also give me the rights for starting my video. Can't start the video as well. So
yes, you can. I had already.
Okay. Yeah, thank you very much. Thank you. Sanish, good afternoon. Are you speaking, Sanish? We can't hear you.
I think most probably there is some issue with his audio.
Yeah, I can hear you guys now. Yeah.
Yes, yes. Now we can hear you. Good afternoon and welcome, Sanish.
Good afternoon, bro.
So welcome, sir. Already we had welcome you. I think there is some audio issue from your side. So may I now request you to have the welcome address.
Yeah, thank you very much. And my apologies for minor delay from my end. It's a great pleasure to be, you could say, be in this meeting. It's a unique meeting from many ways. One, with the increase in clinical research that is ongoing in the country, whether it is of medicines, vaccines, medical devices, even products which are from what we say as the complementary side of medicine, whether it is Ayush related or others. It's a great time to actually be in the research field. And today, I think the topic of discussion is about how to develop or rather how to clinically develop drugs for Ayurveda products. And many a times what comes to everyone's mind is that these products are there in our country and as elsewhere for several thousands of years or even hundreds of years. Why do we need to prove it all over again? And that's from where the origins of reverse pharmacology and how to do limited amount of studies, etc., came into being. all said and done, I think for any science to prosper, it is important for the scientific principles to be followed, as well as in the field of clinical research. It is so very important to have the scientific principles that are followed across the globe to be followed in order for the validity as well as applicability of the research to be, you could say, be confirmed as well as agreed upon by every stakeholder. So in India, ISCR, or Indian Society of Clinical Research, wants to develop in a big way the scientific discipline of clinical research. And this will enable not only the general, you could say, upliftment of clinical research that is done across academia as well as the pharmaceutical industry research. It will also ensure a lot of discussion as well as scientific capability building for research across any discipline, any interpathy discipline as we speak. And today is that day where we will have a good, you could say, selection of topics, especially on how to develop drugs for Ayurveda, as well as some of the clinical development challenges that are there for Ayurveda drugs. So ISCR as an organization, as a society made up of different stakeholders, is very pleased to be part of this conference. And on behalf of ISCR, as well as all the clinical research professionals, I wish everyone happy learning as well as a great interactive discussion. Thank you so much, Shambo, as well as the ISCR East Chapter, Dr. Subhrojyoti and others for the opportunity.
Thank you, sir. Thank you very much. So may I request now Dr. Subhrojyoti Bhowmick, who is the chair of East Zone ISCR, to say a couple of words. Then we will begin the technical session.
Thank you, Shambo, and welcome everyone. Good afternoon. I think East Zone has got a shot in the arm, as we have now Shambo, Dr. Shambo Samajdar as one of our coordinators, and thanks to him that this unique kind of discussion is happening today. And I really want to thank our All India President, Dr. Sanish Devish, who has taken out time from his busy schedule and has given us words of motivation, because Sanish's vision of developing ISCR as the most important capacity building body in the country will come true if we can assimilate all branches of medical sciences. And that is the reason at East Zone we wanted to have this kind of a meeting. We'll have great discussions today from experts in Ayurveda, and we're very happy and honored to have very senior pharmacologist from the allopathic medicine who would be also chairing the session. So let's have a great academic feast, and you will see more activities from the East Zone coming in the coming days. With these few words, thank you very much for this opportunity.
Thank you, Dr. Bhowmick. And we are also really thankful to our President of Indian Pharmacological Society, West Bengal Chapter, Dr. T.K. Mondal, sir. But due to some unfortunate cause, he is not here today. But we are really thankful that IPSWB also are participating in this forum. And also we are thankful to Innovocare E-Academy, who are constantly providing us with technical supports. So now we have to move towards the next session, that is the technical session. And for that, we are really fortunate to have one of the very senior clinical pharmacologists in the country, Professor Urmila Thatte Madam. Thatte Madam was formerly head at Department of Clinical Pharmacology at St. G.S. Medical College and KEM Hospital. So welcome you, madam. And also we have Professor Santanu Kumar Tripathi, who is again a renowned clinical pharmacologist in this country. So welcome, sir, in this academic event. Now we have the talk on phytopharmaceutical development regulations in India. And for that, we have a very renowned speaker in this field, Dr. Shoibal Mukherjee, who is also a clinical pharmacologist and also formerly a VP and head of Asia Medical Sciences Group, Quintel's Research. So, sir will not be present live here. He had given a recorded video for this presentation. So may I request now Professor Urmila Thatte Madam and Professor Shantanu Kumar Tripathi Sir to please proceed for this session.
Thank you so much, Shoaib. I'm wondering, I can't see Shantanu. Is he online?
Yes, I am making him co-host. Sir, you can unmute yourself, sir. Tripathi sir. Welcome, sir. Tripathi sir.
Let me start by introducing Dr. Shoaib. I'm very sad that he's not going to be here. I would have loved to say hello to him. Apart from being a very good friend, he has postgraduate qualifications in medicine and pharmacology with over 30 years of experience in drug development and pharmaceutical medicine. First, he headed the medical and research departments in Pfizer India, Ranbaxy Laboratories, GVK Biosciences, and Quintiles Asia. And he is the founder and the first president of the ISCR, which I think I must congratulate him for, because he showed tremendous vision in setting up this society. And I do remember the first few meetings we used to have when we were just a struggling society. Currently, he is involved in assignments across the pharmaceutical research and medical startup domains as an independent consultant with assignments in pain, tropical disease, biologicals, vaccines, and phytopharmaceutical research. He has been guest faculty for training and doctoral programs at leading medical institutions, and is a member of the research advisory boards and technical committees of various private and public organizations. Dr. Shoaib.
So
Shantanu, I'm sorry I introduced Shoaib. I took over.
Yeah, it's fine.
You were lost to follow up.
No, it is great, but I will be missing Shaibal directly in real time. I'm really happy that it's a recorded version. So in any case, but then it's always great hearing Dr. Shaibal. He's a good friend. When we were doing our DM in PGI, so he has been always a great support to the cause of pharmacology and clinical pharmacology in the country. And yes, he was.
Shambho, I think you should start because we've lost Dr. Tripathi.
Introductions. It's a pleasure for me to be here today, this afternoon. Good afternoon, ladies and gentlemen. Thanks to the chairpersons, Dr. Urmila Thatte and Dr. Shantanu Tripathi for the introductions. It's a pleasure for me to be here today, this afternoon, in pursuit of the Maharshi Charak Jayanti celebrations that are ensuing now. And I'm very happy to present to you the development of phytopharmaceuticals in India, the regulations, challenges, and beyond. So I will be talking mainly about the regulations around phytopharmaceuticals, including traditional medicine and Ayurvedic medicine and plant-based medicine in India. And I will be talking about the challenges. And then I will talk a little bit also about the possibilities of the future and what needs to be done so that India can emerge as a major location for phytopharmaceutical research. So with that, let me go to the first slide. So to begin with, let's look at Indian biodiversity. Now, India has got just about 2.5% of the world's landmass. However, we have about 7% of the world's biodiversity, which means that the the plants and animals, the various species that are there in India, these constitute 7% of the world's biodiversity. These include 4,500 plant species, which is about 11.8% of the world's flora. And not only that, but about 17,500 of these are flowering plants. So we are not talking of, primitive plant species such as algae or even ferns, but of flowering plants, of which about a third are unique to India. So close to about 6,000 species of flowering plants are unique to India. Of these, over 300, that is more than half of them, are considered medicinal plants. So they have medicinal value. And of these, about 900 are commercially traded even today in the marketplace. So there are people who are cultivating or collecting these species from wild areas, from jungles, and selling them in the market. And these are being converted into medicinal formulations. We have a great ancient tradition of Ayurvedic medicine, which has survived even to the present day. And there's been, even after that, voluminous work on medicinal plants in India only in the last century. And there have been notable contributions to modern medicine in the form of ricin, which comes from, or which originally came from the Sarpagandha plant. And the first product, commercial product that came to the market used to be called Sarpasil, based on the Sarpagandha. And we have bromhexine, which comes from the Adhatoda plant, and there's a glycoside called vasicine, from which bromhexine has been developed. So we have some contributions to modern medicine as well. However, there's a contradiction in all of this. There are leading publicly funded institutions we know dedicated to herbal research in India. For example, the Indian Institute of Integrative Medicine in Jammu, which is a large organization which has been there for a very long time. There's an entire ministry dedicated to supporting research in indigenous medicine called the Ministry of Ayush. And there's a thriving domestic market for herbals in India, which constitutes about $750 million. And which is growing quite rapidly at about 14% annually. And yet, India's share in the global market for herbal medicine, which is about $80 billion, India's share is about less than 1%, 0.5 to 1%. And if you look at contributions to mainstream medicine in recent decades, there are none. There have been small contributions that have come in, and they have been written about, but nothing which is path-breaking, nothing which is really eye-catching. And there has not been any essential recognition of Indian herbal medicines or Indian traditional medicine in the area of modern medicine. So what are the reasons? What is the problem? Why can't we make a breakthrough into modern and global medicine with our plant diversity and with so much history and so much tradition in plant medicine in India? So I think, the problem lies in these following points. There is a lack of convincing clinical evidence that can be relied upon for efficacy of these products. Many of us would swear that, yeah, they work well, but to convince the world, you need to have properly conducted scientific studies, and there's a lack of that. There is inadequate documentation of safety as well, and lack of private funding due to intellectual property hurdles. So we know that you cannot get a patent on plant products in India. And as a result of that, if someone puts in money and invests in plant research in India, there is no guarantee that he would be able to dominate the market. There could be other players who would just simply copy that work and do the same thing, use that same literature, and be able to market their product. And if their marketing is better, then all the the investment that goes into the research will not be sufficient to get a return on that investment. There is also no publicly funded scientific program to generate evidence. So now I know that the Ministry of Ayush spends a lot of money on clinical trials on Ayurvedic products and herbal products. However, I have been on committees that that are set up often to kind of evaluate this data and to direct those studies. And I have found that the basic design of those studies is flawed, and also there's no confidence in whether those studies have been done in a manner that fits with good clinical practice, and that we can rely on that evidence, that we can rely on the veracity of that data. There are vested interests of private players in maintaining ambiguity. So now think of yourself as a private company that has developed or is selling in the market certain herbal products. And they're selling to an extent, they're doing reasonably well, you're making a profit on that. Now, if you go in for research that is well conducted, that is well designed, and that is designed to prove whether the product works or does not work, then you have a risk of running that trial and coming to a result that shows that that product does not work. It could be because it doesn't really work, or it could be because the study was not conducted properly for some reason or the other. But once the study is done, you need to publish it, you need to submit it, and it becomes known that the results from this product were not very good. So then you tend to be in a position of losing all that sales that you already had. It's a big risk to take, and people don't want, private players don't want to take that risk. So private investment into research for existing products does not come in. There are hurdles in the form of the Biological Diversity Act that India has, and I will talk on that for a moment in the next slide. And there is a lack of demand for rigor among the scientific establishment. I think the people who evaluate the results and are expected to comment on those results are not looking at the scientific rigor of those experiments sometimes. So let's go on to the next slide. The Biological Diversity Act. This is an act that has been in force since 2003 in India. And this is a kind of act which has a global connection, because biological diversity, it is hoped, and the WHO has been active in this, and other United Nations organizations, they want to maintain biological diversity. And therefore, there is a convention that encourages countries to have an act which helps in maintaining biological diversity. But India has gone much beyond the provisions that are there from the international guidance. And those provisions in India say that there can be no collection of biodiversity, that is, there can be no collection of plants that you intend to do research on without intimation of the state biodiversity board. The knowledge that you gain from doing any research, that knowledge with corporate listed entities requires approval of the National Biodiversity Authority. Application of patent for a biological substance, say a plant product in India, requires the NBA's approval first. You can only apply for the patent if the NBA, the National Biodiversity Authority, gives you an NOC for that. Very large fee is demanded by the National Biodiversity Authority for permission to corporate entities to start doing any work on biological products, which is an additional cost over and above what a company might be ready to spend for the research. And this is taken upfront much before it is known whether that research will even be successful or not. There is no standard fee defined in the Act and rules, and so even this fee is somewhat arbitrary. And often, the National Biodiversity Authority decides on this based on the size of the organization. So if you have greater scope to pay, then you're made to pay that much. And then companies end up deciding whether to spend that money, giving it to the National Biodiversity Authority or put it into some other kind of research where this cost will not be there. So the consequence is that this effectively bars corporate funding for phytopharmaceutical research, and it makes the playing field rather uneven for large corporate bodies, which otherwise do have the money to spend in all this. So that's one big hurdle. What can be done about this? And I'm not talking now of the Biological Diversity Act, but generally about developing India as a center for phytopharmaceutical research. I think what should happen is that there should perhaps be a national program to generate internationally acceptable evidence. Now we know that that commercial organizations will not do it for the reasons that I have just spoken about. But there can always be a national program that is funded by public funds, which are already there and which are indeed being spent by the Ayush Ministry right now. But these programs need to be led by competent people who know how to design clinical trials and how to run them. Apart from just herbal products, which are kind of a plant being converted into a medicinal product and a medicinal entity in the form of a tablet or a capsule or a solution, herbals can also be a great source to generate leads for in the reverse pharmacology kind of manner, so that you identify something in a plant which seems to work, and then you isolate it, you then synthesize it, and then use it or develop it as a synthetic medicine. It has originally come from a plant, but you've probably even modified it a little bit, and that could be a very interesting route. And I don't think we are doing enough of that. Besides, products that have been studied well, products where commercial entities are marketing it, and if they do indeed put in the money to do some research and prove that this works well, and that research is well conducted, and the regulatory authority and knowledgeable persons are convinced that the study is good and it does have efficacy and safety, then whose efficacy and safety is not quite clear, to differentiate it, these products which have been well studied should be reclassified, and they should be given a stamp of approval by the regulatory authorities. And such products should be accepted in hospital pharmacies. They should be used widely in our hospitals, perhaps, because they have already been proved to be good, proved to be efficacious and safe. So there's no reason why not. There should be encouragement of private funding into herbal research by change in the Biological Diversity Act, in the intellectual property rules, and in the pricing paradigm that exists in the country, so that there are incentives for private funding to come in here. Finally, there should be a substantial reward for any new drug that is developed from biological sources and which is useful for the diseases of national importance. For example, an anti-tubercular drug or an anti-cancer drug, or a drug that is for any of the neglected diseases. These should probably be rewarded much more substantially. And one way to do that would be for the government to say that we are going to buy X quantity of such a product if that is proved to be efficacious and safe for these following disease entities. So I'm going on to the next slide here. Let's now look at why are we asking for incentives in this area. And that's because to develop a herbal product is so much more difficult than to develop a chemical molecule as a drug. Let's look at the challenges here. So the development of a new drug begins with the collection of the plant product, the plant itself. And that collection could be either from wild growth, that is, you send out people into the forests, ask them to identify the plant and cut it and bring it back to you. It could be the leaves, it could be the bark, it could be any part of the plant. And of course, permissions are required here from the forest authorities as well as from the Biological Diversity Act authorities. And that is one way to collect. And the other way is to cultivate. So there you then allocate a few acres of land to cultivate a medicinal plant. Now, both of these have great challenges. When it is in the wild and you're collecting from wild sources, from forests particularly, then the people that you send out there, these are collectors, these are manual laborers. They may not even be educated in how to identify the plant. You have to train them how to identify it. And then they are usually remunerated based on the quantum of collection that they have done. How many kgs of the product has been collected. Now, so there is an incentive for these people to collect not only the relevant plant, but possibly also along with it, other things that are not really this plant, contaminant plants out there, herbs or leaves or whatever that is, because that just increases the volume of what they have collected and ends up in being more remunerative to them. So that's one challenge. And the challenge, of course, is also that all this has to be collected and immediately transported to a processing place. So if you have a collection drive for a period of 3 or 4 days, every day that collected material has to be transported, because otherwise the material will get stale. So if you store plant product, the moment you cut it, it begins to die. Plants obviously are living cells, and they begin to die the moment you have cut it. So you can't keep that plant for very long. It will grow fungus, it will grow other contaminants, and it might change in nature, and its medicinal properties might also disappear over a period of time. As far as the cultivated material is concerned, there are other challenges. When you cultivate, you need to allocate a particular amount of space to it. And then the remuneration from that, the cost or the price at which you are able to sell it should be higher than the price you would get if you allocated that same amount of land to the growth of, let's say, potatoes or wheat or whatever else. So that's another challenge. There are more challenges, and I won't go into all of those right now. We don't, I think, have the time. But the transportation is the next next challenge. You have to immediately transport it, and it could be a short haul transportation because your plant then needs to be located close to the areas from which the plant material has been collected. But if it's a long haul, your plant is a national plant somewhere there in maybe Hyderabad, and you're collecting the plant material from the hills in the Himalayas, then that transportation cannot be perhaps by trucks or by the road, because it takes a lot of time, and during that period you have to either refrigerate those trucks or you have to do something to make sure that the material does not degrade. Once it gets to the processing plant, then there's a whole list of things that need to be done there. The first thing is sorting, because there will be a lot of contamination. And so what you want, the parts of the plant that are required have to be sorted out from the parts of the plant that are not required, and any contaminant plants that have been collected in the process. The washing is very important. When I was working on a plant product not too many years ago, we discovered that that plant that was collected from near the highways had a higher contamination of lead in them in comparison to plant that was collected from fields which were further away from the highway. And we ultimately realized that this was because the trucks that ply on the highways release exhaust which contains lead, and that lead is in the air, and then that settles down on the leaves. And when you do this, there's a higher contamination of lead, and it could be any other heavy metals as well. So that is a challenge, and it has to be washed properly, and maybe sometimes other means have to be put in place so that such contaminations are not there. Then the material has to be crushed, it has to be dried, it has to be extracted. The active principles have to be extracted from that. Often extraction is the process by which this whole chain works. And then it has to be purified, because there might be residual solvents in there, there might be other contamination. So one has to look at the purity of it. And then, of course, like any other chemical substances, it has to be formulated either into an oral solid, which is usually a capsule in the case of plant products, or a liquid oral, or a topical medicine. It has to be then labeled, classified, packaged, and then distributed as per the label. And the distribution chain will have to follow the regulations. So if it is classified as a phytopharmaceutical product, then it can only go to pharmacy shops. While if it is a herbal product of the Ayurvedic nature, then there are many other outlets from where it can be sold. Okay, so let me now go to the next slide. Now that we have talked about Ayurvedic products on the one hand and phytopharmaceuticals, which are not Ayurvedic products, on the other hand. So let's look at the differentiation between the two. A plant product which is mentioned in the books, the scriptures that are listed in the first schedule of the Drugs and Cosmetics Act. These are, the Charak Samhita, and we are today celebrating the Charak Jayanti. So the Charak Samhita, and there are other a list of many books, traditional books that list Ayurvedic medicines and how they are to be prepared. If your plant is listed in these, then it's a yes, you can go to the route of Ayurvedic medicine, and then it becomes much easier for you to get a regulatory approval to market this product. And there are certain rules. If there is no change at all, you are using the product in exactly the same way as it is described in the scriptures in the published, then you don't require too many permissions. You have to only submit published literature for that, and that is sufficient. If it's even a new formulation, even then only published literature suffices for an approval. If it's a new indication which is not described in the scriptures, then you require a proof of effectiveness, so you will have to do a clinical trial. If the ingredients are as per text, again, there has to be a pilot effectiveness study. And if it contains any ingredients which have safety concerns, then a safety study would have to also be done. And if it's an extract, if it's an aqueous or a hydroalcoholic extract, then there are no further mandatory requirements. On the other hand, if it is an extract in alcohol, which is greater than 50%, then further safety and effectiveness studies have to be done. It is all there in the Drugs and Cosmetics Act and in the new NDCT rules. So I will not describe it any further here. You all can all look into it. But if your product is not mentioned in any of the traditional Ayurvedic scriptures, but it's a plant product, then it has to be developed by the phytopharmaceutical route, which is very similar to the other small chemical entities route as far as the Drugs Controller's Office is concerned. And you have to submit this to the Drugs Controller's Office and not to the Ayush Ministry. And it will be evaluated by the norms of the CDSCO as a phytopharmaceutical. And we'll talk a little bit more about that. These rules are described in the New Drugs and Clinical Trials Rules 2019, NDCT, NDCTR to be precise, 2019. And they are there in Table 4 of Schedule 2 of these rules. So let's look at NDCTR 2019, Table 4 of Schedule 2. The data that you have to submit has to be in two parts. Part A is general information that you can obtain from published literature. And these include the name, the description, and the claims you are making of this botanical product. You have, if it is already a marketed product and you want it to be to go through the process of review by the regulatory authorities, then the current volumes of sales and the safety information from current use has to also be submitted. Any published reports from traditional medicine and recent animal or human studies, if they are related to this product, have to also be submitted. And monographs and reports of pharmacodynamic and adverse effects, if they exist, have to be submitted as well. So this is part A. And the part B is specific data that you would have to generate on the product that you are intending to sell. And these include the CMC data. CMC is the data on the quality of the product. So this is the raw material, the drug substance, and the drug product. And then you have the safety data that comes from the preclinical trials. And then you have the efficacy data that also comes from clinical trials. And the regulatory status. So some administrative data has also to be submitted. We'll look into this on the next slide. So what are the CMC requirements here, the quality requirements? Here, there is a great deal of characterization that you need to do on the botanical raw material. So the plant that you have collected has to be characterized in macroscopic form. That is, you have to say what is the shape of the leaves, how do you identify the leaves, if it's the leaves. If it's the bark, then you have to give some macroscopic identifiers for that plant. Microscopic characterization has to be there. That is, how does it look under the microscope? Genetic characterization, if that has been done, has to also be provided. And some of these rules are required if you want to globally market it as well. The US FDA also has the same rules. And then the collection specifications, that is, when is this plant supposed to be collected? What time of the year? If it is to be cultivated, then what time of the year has it to be harvested? What time of the year have the seeds to be sown? And so on and so forth. Then the botanical drug substance, that is, this was the raw material. Now you come to the drug substance, that is, you've already done the processing and the extract is out there, and that's called the drug substance. It's equivalent to an API for a small molecule. And you have to identify phytochemical markers. So here is an extract. Unlike a chemical product, which is only one ingredient, here you have, it may be a mixture of many ingredients. And some ingredients may be in greater quantity, other ingredients might be in lesser quantity. You have to be able to identify, to be able to say that batch to batch variation is not there, that it is the same product this year as it was in the last year. You have to be able to identify some phytochemical markers. That is, 2 or 3 or 4. And the requirement of the DCGI is at least 4 phytochemical markers have to be identified. These are usually those substances that when you run on an HPLC, these give the biggest peaks. And then you've got to do mass balance studies, that is the total quantity, how much of each chemical is contained in this total quantity. So anything that is more than 1% has to be quantified, and anything that is less than 1%, you say that the residuals. And then what residual substances are there? Are there any harmful residual substances? For example, residual solvents are usually an important part to eliminate here. After that has been characterized, the drug substance, you've got to document the processing steps that were taken to get the product out. So how was the sorting done? How was the cleaning done? Was the water that was used, was it distilled water or normal tap water, and so forth. And the drying process, how long was drying done? Was it done in a drying chamber or was it done under the sun? The extraction process itself, what were the solvents used? How long was the mixing happening, etc. The desiccation, how long was it dried after the extract was obtained? How was the solvent removed from the mixture, and so forth. And the biological activity studies have to then be documented and submitted. These could be based on fractionation, that is, you would then want to purify this extract further to get down to one ingredient or a very small number of ingredients in which one ingredient is predominant, and see whether in animal studies it has activity or not, or in vitro studies for that matter, and identify the active fractions. Sometimes this is possible, sometimes this is not possible, and it is not mandatory to do this, but you should at least try to do it and be able to identify the chemicals that are responsible for the activity of this product. And then stability studies have to be done on the drug substance as well as the drug product, biological stability, chemical stability, and physical stability, all three of them. So these are the CMC requirements, and you can see how much more difficult this is than it would be for a chemical product. There are some permitted deviations. In the Indian law, non-clinical pharmacokinetics and toxicokinetics is not mentioned. So animal pharmacokinetic studies do not need to be done. Reproductive toxicology is also not mentioned except in the context of a product which would be used in pregnancy, where you have to do teratogenicity studies, and that's the only term that they use in the NDCT rules. Carcinogenicity studies are also not mentioned anywhere. As far as the clinical testing is concerned, abbreviation and modification or relaxation can be availed in the conduct of clinical trials for a drug that is already in the market, has been in the market for more than 5 years, and where there is adequate published evidence of safety. So some relaxations are there. Now let us look at some of the other challenges that we have in comparison to chemical products with biological products, with plant-based biological products. What problems we can sometimes face. Conventional pharmacokinetic measurements are rarely possible with plant products, and because of a variety of reasons. The active ingredient often is unknown. So you have a mixture which is an extract from a plant, and it is thought that this is effective, and maybe you've also done a clinical trial to show that it is effective, but you don't know what in that mixture is the active ingredient often, because it has not been possible to purify that active ingredient fully, and also because the biological activity is usually not an activity of the single compound in that mixture. It may be the activity of a combination of chemicals that are there in that mixture, and not only that, there might be enhancers of that activity within that mixture, or modifiers of that activity, or dampeners of that activity, some ingredients which stop a particular side effect, which would happen otherwise if you were to use this product, this chemical on its own. So pharmacokinetics really does not tell you much, and it's very uncertain. The measurement of chemical markers is also an unsatisfactory compromise, because the chemical markers that you identified may not be the active ingredients. There is no rational basis for the choice of the marker to be measured. It's only those chemicals that are in the highest concentration that you identify as one of those 4 chemical markers. But it is not necessary that these are the active ingredients, and it is also not necessary that all 4 of them are active or only 2 of them are active. You know, it's a mixture that you don't know too much about, and it's very difficult to know too much about. There's no guarantee that the marker pharmacokinetics will correlate with the pharmacokinetics of the activity of the drug. And the marker measurement may be only necessary to rule out accumulation of some inactives. So one way out of all of this is to measure biological activity. So what you do is, instead of measuring the quantity of a chemical in the blood to mark the pharmacokinetics, you measure the biological activity. You take out the blood itself, and then you use that blood or the plasma in an in vitro assay to see how much of biological activity it has, so that if you're wanting to block a receptor, then try to see whether how much of the receptor blockade is taking place with a certain quantity of blood over a period of time. And you might find that you can get a pharmacokinetic profile of increasing activity and then decreasing activity over a period of 12 hours or 24 hours. And that gives you, this is what was done, for example, in the early days with insulin or with other biologicals, because we did not know how to measure them. So in vitro assay has to be adapted. The challenge here is that any assay has to be adapted to the matrix, that is the blood. So the blood might contain certain interfering factors which do not allow you to measure that activity. So these are some of the challenges of pharmacokinetics. Some of the challenges also relate to dose range finding. For example, for a chemical product, you base your doses on the pharmacokinetic profile. You know that here is the substance, this is its peak plasma concentration, this is the half-life, and therefore based on that half-life, if you give it twice a day, that would be sufficient. In this case, as we said, the PK profile itself is an issue. So therefore, the dose range finding becomes a bit of a problem. So you might need to define the dose based on the activity in blood that you get and the pharmacokinetic profile that you deduce from the activity profile. The PK profile of other key ingredients must also be taken into account. So if there is another ingredient which has lower activity but has a much longer half-life, then you might have to modify the dosage accordingly. A wider dose range would have to be tested if activity and pharmacokinetics is not known. The maximally tolerated dose in healthy volunteers would be a key dose level marker. So you have to get to an MTD. An adaptive approach may be required for the choice of dose because of all these foregoing challenges, because you don't know what the range of doses might be. You take an adaptive approach, you give a lower dose, then you see the effect of that and then decide the next dose to be given, and so forth. Dose finding may require a prolonged sequential testing of doses. So there is really quite a bit of challenge in the phase 2 of the clinical design. Often, because for most of the diseases, there might be active and well-known chemical drugs, you might have to adapt an add-on design for the design of trials for plant products. Often it requires a large sample size because the incremental effect on the patient might be small, and to be able to measure that small incremental effect in an add-on design, you require a much larger sample size. So this becomes much more complex, and this becomes much more expensive. Placebo availability is also a problem, because often masking is very difficult with plant products. Often the volume is quite high that you have to consume. So placebo availability, while it's a challenge, unless you have placebo, it becomes difficult to do a well-controlled study. Blinding may be an issue, particularly for large volume liquids. Objective assessor blind evaluations may be required, and compliance assessment may be important, because the taste and the frequency of plant products often comes in the way of compliance. You have a plant product which has to be taken 6 times a day, and patients will probably not take it that frequently. So you have to be able to measure compliance, and because of bad taste or something like that, particularly if it's a liquid product, then compliance again becomes an issue. And then planning for these trials is not easy. There must be accurate forward planning of new botanical product trials, and that's critical, much more critical than chemical products, and I'll tell you why. So if your dose for a particular disease is 100 milligrams, 100 milligrams per day, and suppose you require 100 doses, so 1,000 doses. So let's say over a period of time, over a 1 month period, you require 100 grams of the extract to be given to a patient. The trial is, let's say, a 1 month trial, and you require 100 grams per patient. Then where do you get that 100 grams of extract from? You get it from a dried plant where you do the extraction. And in this extraction process, 1 kg of dried plant will result in possibly 100 grams of the extract. So actually for 100 grams of extract, you require 1 kg of the dried plant. And we know that plants are not dry when they are harvested or when they are collected from the jungles. So often, 60%, 70% of the weight of a plant is only water. So the moment you dry it, it reduces your in size. So 1 kg of dried plant is actually 3 kgs of cultivated plant. So to get 1 patient dosing, you require 3 kgs of cultivated plant. 100 patient study will require 300 kgs, and 1,000 patient study will require 3,000 kgs. This is the volume, first of all, that you have to determine of how much to be cultivated. And then how much land you require for the plant to be cultivated on, for this quantity of plant to be available to you, you must decide and cultivate that amount of land. And also, it will take some time for the plant to grow. So you have to sow the seeds 6 months or 9 months or a year before you are going to harvest the plant. So you must know all of this 1 year, 1.5 years in advance of how much land you want to cultivate, how much cultivated plant will come out of it, what is the yield of that plant, and what is the seasonality, in which season are you going to extract, cut it, collect it, and then process it. Ideally, therefore, the trial sample size and the dose that you're going to be using in the trial should be known to you at least 1 year in advance, sometimes even longer than that. So, I think I come to the conclusion of my presentation here. My effort has been to demonstrate to you what the potential of herbal products is in India, how our biodiversity is high. We have a tradition in herbal product development in this country, and study and research on herbal products in this country, and yet we are not doing too well at the present time. There are some things that need to be done going forward, and it's not easy, it's challenging, and some of the challenges come from the rules and regulations that have been put in place, which probably need to change. So in conclusion, we have a huge scope for phytopharmaceutical clinical trials in India. Single center academic trials can probably be undertaken at a low cost. However, placebo availability is a must for an acceptable design. The ambiguity in efficacy of widely available herbal products that are already there in the market can be resolved by a program of clinical testing and publication. Funding for clinical development of new phytopharmaceuticals is constrained because of lack of publicly funded programs and the absence of incentives for private investment into research. However, there is great scope of lead generation from our herbal heritage, and which is also constrained by regulatory and intellectual property issues. If these issues can be resolved, then I think there is a great future for phytopharmaceutical research and herbal drug product research in this country. With that, I would like to thank the organizers for giving me this opportunity to present at this forum. I will not actually be there to present this live, so this is being recorded and you will hear a recorded version of this presentation. Unfortunately, therefore, I'm unable to take questions. I would have loved to do that because the questions are the key at the end of a presentation to clarify matters. However, I don't have the opportunity to do that this time. Maybe next time we will do that. I would like to thank once again the organizers and the chairpersons of this session for giving me this opportunity. Thank you very much.
Thank you, Shoaib. I mean, he isn't here, so he won't be hearing me. But may I request Shantanu to summarize, if you like, or there are no questions.
Yeah.
So he's not there to take questions, but any comments?
Yeah, there have been I think, couple of comments or questions in the chat box, what I saw. But then I must say that was a very thought-provoking presentation, highlighted the current challenges. The list of challenges are quite huge for that matter. And I was just wondering if it has been put in the New Drugs Clinical Trial Rules 2019. And given the fact that, as Dr. Shaiwal has pointed out, that there might not be too many enthusiastic takers from the industry for that matter, and it is public funding that we are expecting. And in the current model of public funding in India, let me put a question to Dr. Urmila, because I know Urmila also has a huge interest for almost a couple of decades in this area of herbal phytopharmaceuticals research and all. So what do you think that because Shaiwal is not here, so what is the prospect of this, given the huge challenges that has been very rightly pointed out by Dr. Shaiwal? Once it has been put in the regulation in 2019, how much we have progressed in the last, say, couple of years, although most of the time is also was pandemic time. But then is there any hope to is there any reason to be hopeful about really some worthwhile phytopharmaceutical product to be licensed in near future, number one? And second, that phytopharmaceutical licensing, as we understand then, will be from the mainstream drug regulatory agency of CDSCO. We know of Ayurvedic licenses. We know of the synthetic and biological licensing by the DCGI. But what about phytopharmaceutical licensing, phytopharmaceutical product licensing? It appears that it will also by the CDSCO and DCGI. But then whether it will, these are the two questions. Dr. Urmila, would you like to comment on this?
Yeah, I just want to say that I don't see any product which is going to shake the world as yet, at least. But I'm always hopeful because but you know, the challenges that Shoaib mentioned are all very valid. And the most important challenge is that there is very little return on investment because of the inability of anybody to take a patent. It
Right.
is extremely expensive to do research, which will give you those markers and things like that. So the run of the mill Ayurvedic industry is just not interested. They are not interested because the return on investment is just not going to give them their returns.
Return on investment, right?
Yeah, so I think that, yeah, so we really have to be very careful. Should we look at these questions? Is there a separate window section at CIDESCO? I don't know the answer to this. Shantanu, do you know?
There,
If there is
I
a
have heard.
separate-
I have heard there is a separate window with, I think, Dr. Katoch, DC Katoch,
Okay.
who is looking after this.
Ayush. Okay.
This is a separate. In fact, what they have done, maybe rightly, that they have dissociated with from Ayush. Phytopharmaceuticals will be looked after by CDSCO and DCGI, while the Ayurvedic products are going to be looked after by the Ayush Ministry and the
Yeah,
Department of Ayush.
that's separate.
That's the thing.
The second question is, should such studies be reviewed by Ayurvedic institutional committees or any other institutional committees? So you have to actually, whichever the center is, which is doing the study, should have an ethics committee to review this. Their ethics committee should review it. And it is a good idea to get it reviewed, the project, to get it. And one more thing is that the ICMR guideline says that you have to have a collaborator from that stream of medicine. It's Ayurveda or if it's homeopathy or whatever, that person should be a co-investigator. And the responsibility of dispensing the medicine goes to that person and taking care of the patient as well. But I would like to say one more thing. What is your opinion about the black box research methodology for Ayurvedic research? Because modern methodology cannot, yes, this is what I feel is extremely important. And I've been saying that for a long time, that we need to develop research methodology. I don't think that this gold standard of randomized controlled clinical trial can be just cut and paste and applied to Ayurvedic medicine. It can't be, because there are so many issues in Ayurvedic medicine that we don't even know of, that we don't even talk of. And we've done this research in the past, in my last avatar, when we have shown different prakritis to have different responses in terms of cellular responses. And so, you know, these prakritis prakriti that they talk about in Ayurveda does influence the response to your drugs as well. So it is very crucial that we develop a research methodology so that we are able to develop a proper system that will evaluate the drugs in a better way. For any clinical trials, herbal, we always get the EC approval. Okay, this is the response. For example, same drug like Kuta just given at low dose. Okay. I don't know, this was just.
No, I think what one thing
there are
they
no
have
more
done.
Questions.
I think what they have done, at least one thing they have done, they have dissociated the Ayurvedic products from the phytopharmaceuticals, and they have defined phytopharmaceuticals, and they have also mentioned that phytopharmaceutical research will be as good as the new chemical entity research or the synthetic chemical research.
Yeah.
Okay. So that way, the application of the prakriti, etc., that goes well with the Ayurvedic products rather than the phytopharmaceuticals.
I agree with you, but I'm talking about generally, you know, when why is there no product available? One of the reasons is that we don't have anything that is
absolutely
earth-shattering in this, because our research methodology needs to be improved.
One take home could be, it is much more challenging. Okay, and we need to develop its own different, you can say, SOPs for the phytopharmaceutical product development, which will be distinct from the synthetic or the biologicals that are prevailing now.
Yeah.
So with this, I think we can possibly conclude the session with ample thanks to Shaibal, in absentia, and we thank the organizers for giving us, myself and Romila, the opportunity to chair this wonderful session. Thank you very much.
Thank you very much. Thank you.
Thank you, sir, and thank you, madam. Because of you, this session become also more lively. Also, we are really thankful to Dr. Shoibal Mukherjee, sir, for this wonderful deliberation. So we have to move forwards towards the next session. And for that, we have two esteemed chairpersons, Dr. Shandip Mukhopadhyay, who is the Scientist E, Medical and Deputy Director of ICMR, National Institute of Cholera and Enteric Disease, Kolkata. And also we have Dr. Subhrojyoti Bhowmick with us, the chair of Indian Society of Clinical Research East Zone, and also he is the clinical director of Peerless Hospital and BK Roy Research Center, Kolkata. So welcome you, both of you. So we have a short video message from Dr. Shandip Mukhopadhyay. Please start that video. May I request Rohan to please start that video. Rohan, are you there?
Yes, I'm there. Just a moment.
Respected faculty,
Is it visible?
Yes, yes.
Hello? Okay.
respected faculties and dignitaries, it is a great pleasure to know that ISCR East Zone is going to organize this Charak Jayanti today. We know that Ayurveda was there, Ayurveda is there, and Ayurveda will be there in the future also. The root of Ayurveda could be traced back to ancient India, and hence we always feel very much comfortable with Ayurveda as it is linked with India. However, good research is required, and it is a necessity to keep Ayurveda contemporary as well as to serve many more people who are suffering from different types of diseases. So, good research is not only about the efficacy, but research with safety endpoints are also required, because anything which has got some effect must have something undesired. And many of the Ayurvedic products are crude extract from the plants, and therefore must be containing something which is not desirable. So all these things can make the product in some places as a safety issue. So safety monitoring must be there for all the Ayurvedic products. I'm happy that in this program we have amongst us Sri Achinto Kumar Mitro with us, who is not only an eminent Ayurveda physician, he is also the officer in charge of the Regional Ayurveda Research Center in Ranikhet, Uttarakhand, under the Government of India. So I'm sorry that I could not be present here today live because of some other commitments. So I wish you all the best, happy learning. Thank you.
Yeah, so now may I request Dr. Subhrojyoti Bhowmick to start the session, please.
Thank you, Dr. Shambo. And without wasting much time, may I now request Dr. Achinto Mitro, who is amongst us and is the officer in charge of Regional Ayurveda Research Institute, Ranikhet. Sir, please start your session, and then we will have some discussions post your session on the question and answers which come in. Dr. Achinto Mitro, please.
Okay, thank you. May I audible?
Yes, yes, you are. You're audible.
Good afternoon. Namaskar to everyone, and respected chairperson of this session, Dr. Subhrojyoti Bhowmick and Dr. Shandip Mukherjee, and other respected experts like Dr. Tripathi, Dr. Urmila Madam, and participants and my co-speakers. I'm extremely happy that and also thankful to the organizer, that is the Indian Society of Clinical Research, East Zone, and Indian Pharmacological Society, West Bengal Chapter, for conducting this webinar on the theme Research in Ayurveda under the celebration of Maharshi Charak Jayanti Mahotsav. And also you all aware that Government of India also celebrating Azadi Ka Amrit Mahotsav at the rate of 75 years of independence with a lot of activities. So today's topic is the safety monitoring studies in Ayurveda. And I will, because I am in the 17 years in the Research Council, since Kerala to Kolkata, and now I am running here. And also we are also part of the pharmacovigilance program. So let me discuss about this, the strength and opportunity and threat of this Ayurvedic system of medicines in safety monitoring of the study. As you know, this is Ayurveda, very ancient system of medicines, and also our nation pride. Being an Indian, we can feel proud that we have own system of medicines. And Ayurveda is definitely given the priority in the drug safety. And if the exact term is not mentioned, but the essence and the flavor is also there in all the classical texts, like Charak Samhita, Susu Samhita, Bhagavad Samhita, like that. And nowadays also we are considering under this pharmacovigilance program. The major goal of this pharmacovigilance is that, in Ayurveda there is a Ashtanga, 8 types of disciplines. And there is a very important segment is that Dabbaguna Vigyan, which is Ayurvedic pharmacology, Vaishajya Kalpana, that is the pharmacy or pharmaceutical sciences, Agat Tantra, which comes under the toxicology and forensic medicines, and definitely the treatment of therapeutics, which is called Kaya Chikitsa. One study I have seen that in the USA, NCCM, that is so that 7.5 lakhs people of the US, they are taking this Ayurveda, and also 1.5 lakhs people they are using in the last 12 months. Means I like to mention that the demand of Ayurvedic medicines and as well as the herbal medicines is growing very fast. It is reported that 7.5% per year growth is there. Nowadays, the safety monitoring Ayurveda is the highest priority by the Government of India, Ministry of Ayush. There is a scheme is going on since 2017-18, the Central Sector Scheme on Pharmacovigilance for the Ayurveda, Siddha, Unani, and homeopathic drugs. And GCP of Ayush on Ayush is also now available. Compliance of ethical clearance, ethical approach is also mandatory, and you have to have CTRI registration, which is maintained by the autonomous body of under the ICMR. This is now mandatory. And in every clinical trial, we have a separate segment to reporting for ADR, AD, if any. Next. And there is always a misconception. When we started also pharmacovigilance program in 2007-8, there is among our physicians also, why you are doing this kind of things, pharmacovigilance? Because our drug is safe, because our drug is based on natural products also. But if you see in Charak Samhita and other Samhitas also, they have given more emphasis on this, the safety of this drug delivery. And mainly depend on this, the qualities of this plant product or whatever medicaments we are using for the treatment purpose. And another important thing is this, Ayurveda is a personalized medicines for which it is a little bit different. The basic principle is different from the conventional system of medicines. So medicines may vary according to the constitution or prakriti of the patients. So these kind of things we have to keep in mind during this treatment of the safety monitoring of these Ayurvedic medications. And interestingly, there is the Ayurvedic the Ayurvedic medicines also having this metal minerals also. It is not that roughly you can say that 85% we are using the plant products. Next slides. And mostly 15% raw material we are getting from this metal, minerals, and marine products also. So this is also very challenging sometimes because we are using these metal minerals in the form of after incineration as a form of bhasmo. So that is also challenging area nowadays, and most of the popular medicines in Ayurvedic classics like Aradhavardhana Bhoti. Next slide, please. Also have this plant product and also metal minerals and marine products also. So it is the complex products we are using for nowadays also since 1,000 years. That is also the presence of the metal toxics and their compliance. However, nowadays the GMP compliance, we can say 100%. However, how much they are following the traditional methods of preparation, that is also very important thing, and also create some hazardous effects in the practice also. Now, Ministry of Ayush has developed a dedicated portal that for safety monitoring of Ayurvedic medicines at the doorsteps of the physicians, and that is ayushsurakha.com. You can go anytime and you can see the detailed information you will get from this portal. And also there are so many positive things is that anybody can submit the ADR/AD through this portal also. Now, there is a very old Drug and Cosmetic Acts 1940 and Drug and Magic Remedies Act on the OA, that is the 1951. These acts are now amendment, and recently one law enforced, that is the 170 prohibition of advertisement of Ayurvedic, Siddha and Unani drugs, which is very, very important, I think, and there is a chances of impose of the judicial punishment of these victims. And new drug and clinical trial rules 2019 has also implemented. So this is the recent achievements of the Ministry of. As you know, this when the poison become a good medicines, Yogadapi Bisham Tikna Muktam Bhesaja Bhavet, Bhesajam Chapi Durjuktam Tikna Chamaveda Bisham. So the meaning is that basic principle is that a poison can become a good medicine if you use properly with a skillful administration. But if you misuse the good medicine, can be also a poison. So that is mentioned since 1,000 years ago. So now the need of drug safety is definitely the priority. And if you see that the ancient times, even if you back to the 100 years, the Vaidyas and Kabiraj, they are used to prepare the medicines, they are cultivating, they are collecting from the forest, they are preparing their products, and they are dispensing also by. So they are controlled totally in the single hand. Now nobody is preparing, it is not possible in the modern era. Now home scale product become a large scale product by the manufacturing of the different companies. So that is the changing scenario of the present day. That's why drug safety is very, very important. And if you see that Ayurvedic Sangitas, we have two types of medicines, like classical formulations, which are using in the 1,000 years, and some medicines, which is proprietary medicine by the companies. So we have to look at this all the, because ministry has given some gazette notification that if you have a classical formulations you're using, and also having that SOP and by the Ayurvedic Formulary of India or API of India, you don't have to go for the clinical, the toxicological studies. But if it is a new drug, you have to have the preclinical study before entering the clinical trial. So there is a GMP certified Ayurvedic pharmacies in India, and the pharmaceutical products reach value of 5,15,500 crore in 2021. So it is expected that the growth is 19.78%, and it will reach 2027. It will 15,36,900 crore. The market is gradually growing, so the safety aspect should be taken care. Next slide. So if you see that the little bit infrastructure of the Ayurvedic system of medicine in India, that is a 170 undergraduate college and 64 postgraduate college, and there is a practitioners now is about the 7 lakhs in the country. And there is many challenges of Ayurvedic medicines focus that pharmacovigilance program. That's why it is very, very important, because there are about
Dr. Mitro, we are not able to hear you.
is it okay?
Yes, now it is fine. Yes.
There are 8,000 manufacturing companies in the GMP certified companies in the country, and about the 33,000 hospital and dispensaries in the government sector. I'm telling the dispensaries available. So detection and adverse reaction to the Ayurvedic medicines is a very, very important thing, and under the central sector scheme of pharmacovigilance program. So concept and terminology I have mentioned, it is not there in the like the ADR, AD or signal detection like that. But Ayurvedic syllabus now they have included this pharmacovigilance, and method of study, drug safety problem now evolved gradually in Ayurveda. If you compare to the 20 years back, there is nothing. But nowadays, we are trying to aware these students and as well as the practitioners. And although this information related to medicine exists in the Ayurveda, it is not easily accessible, because our approach is the multi-drug, multi-therapy, and multi-drug prescription. So, and also each and every drug having so many molecules. So it is not so easy to accessible. Signal detection is very difficult because the inherent belief about the safety of Ayurvedic medication leading to lack of reporting, collection of reports also nowadays is also challenging. So more and more ADR, AD is getting difficult to do that signal detection. Patients often use medicines from different system of medicines concomitantly and simultaneously. You can say if he's a diabetic patient, he's giving modern medicines along with some home remedies, some homeopathic medicine, some Ayurvedic medicine simultaneously. So this is also very difficult to do the causality assessment. The lack of quality assurance control still is a challenging. If it is I have already told the GMP certified company 100%, but the monitoring part is very weak by the state drug control. Till it is a very challenging. And manufacturing and selling Ayurvedic drugs, because the small scale selling unit is larger. So that is also very, very challenging in the vigilance aspect to catch the people, those who are doing a mistake. And also important thing is that nowadays in the market, and if you open the newspaper, television, so many spurious drugs advertisement is there. Here I've given that one company during COVID time, ministry has given some advisories that if you want to boost up your immunity, you can take the turmeric, ginger, clove like that, which is available in your kitchen. One company, they have made the bread with these herbs, and they're claiming if you take this bread, your immunity will be boosted up and you will be free from the COVID-19. So this kind of claim also misleading and defaming this system of medicines. In Calcutta also, one reputed sweet manufacturing company, they have produced immunosweet sandesh, so which is having a lot of 17 herbs, and if you take, then it will be you can fight against the COVID-19. So like that, this is going on in the market also. Next. So assessment of the adverse drug reaction to Ayurvedic medicines is a very, very important thing. Information related to adverse effects, whatever mentioned in the classical text, it is a very scattered. I have told it is somewhere it is written the Paguna Vikan, Rasastra, Bhaisajya Kalpana, Agattantra, Kaya Chikisha, like that. So that is also, we are trying to gather in the same, that's why the portal is formed, and many publications are not in a peer-reviewed journal because there is no dedicated journal for Ayurvedic system of medicine. Very few journals are there. So publication is also because we don't have enough data to publish. Whatever we have started since 2018, I think data is now gradually generating. Most Ayurvedic formulation is the multi-ingredients, fixed dose, like that. We have worked on this one system, on medicine, that is Sarnabhasma. We have studied that it is classical dose. There is a sarnobhasma, but it is having also organic material also, and also having some efficacy of the organic part also. So it is a multi-ingredients. I have already told we are using the plant product, metal, mineral, like that, marine product also. Additionally, there is a confounding factors of the patient is often receiving Ayurvedic medicines with allopathic. Mixotherapy still is also challenging. If you have any ADR or AD for the causality assessment, it is really challenging. Next. So pharmacokinetics and toxicokinetics is very difficult. Most of the Ayurvedic formulation, we don't have this data or that study. Dose-related response also rarely measures. Sarnobhasma, we can say we have studied on the sarma also. That is the classical dose is 125 mg per day. But we have seen that the 6 mg is enough. So 6 mg is equally effective if you give the 125 mg. So this kind of study is very less, and really you are able to do that de-challenging and re-challenging, because we have a lot of formulation, thousands of formulations. So option is there much more. So always doctors prefer if there is any adverse reaction or a kind of side effect, they're changing the drug immediately. Their very attitude is not there to be a re-challenge of this drug, which is a vital parameter for drug safety monitoring. And one of the most challenging aspect of this, of the lack of expertise in the causality as analysis. However, we are now forming the committee of the multidisciplinary approach by which we are trying to solve the problem of the causality assessment of this NAD or AD. Next. That is the advanced drug reaction. The Ayurvedic medicines, the practitioners, because in the modern medicine like that, this pharmacovigilance program is going on. However, the Ayurveda is not a part, but herbal medicine, botanicals, phytochemicals is the part of their part, not in ICE system or Ayurvedic system. So Ayurvedic practitioners are less aware. However, where every center, pharmaceutical centers or the intermediary centers, they are conducting so many awareness program among these students and all the practitioners. And but it will take time. Proper drug information of the Ayurvedic drugs, including both classical and proprietary formulation, not available easily, because we are only depend on the Ayurvedic formulary of India or Ayurvedic pharmacopy of India, where the whole the formulation is not covered, and patients are not adequate aware. Sometimes they are believing that Ayurvedic medicines have no side effect or adverse reaction. If anything happen, it is due to maybe other cause. So they are ignoring sometimes this if anything happen. And education in the modern and Ayurvedic medicines, both the undergraduate and the postgraduate is very, very essential on the pharmacovigilance. But that both conventional science and the ICE system, both they should know each other of these medicines. I think it will be very valuable, and now present day it is very, very effective. The Ayurvedic pharmaceutical industry, it is not included till now in the pharmacovigilance program, but they are the major player of this pharmacovigilance or the safety drug delivery system. So that should be included, I think, and availability of Ayurvedic medicines is very, very variable. And there are so many books, more than 100 books describing different formulations and medicines containing 1 lakh formulation. I can mention one book in Bengali, it is written Ayurveda Sangraha, which is having 1400 pages and thousands of formulations. So this kind of information we have to ready to hand. That is also very challenging for the safety drug delivery system. Next. So it is the standard methods, because Ayurveda, they have so many formulation in the particular disease, and also it may be varied person to person. So if you are knowing that this drug is causing harm, then we can change, we can avoid, by which we can prevent the adverse drug event. That is the, I think, strength area of Ayurvedic system of medicine, because if you are considering one particular disease conditions, we have a lot of formulations. So option is there, and we can avoid this if it is reported there is an adverse reaction of this particular drug. Next. So in the pharmacovigilance program on ASU and AHE system is going on 2017-18, and where the All India Institute of Ayurveda, New Delhi is the national coordinating center. Under his every system have their intermediary center. In Ayurveda, there is a 2 intermediary center. One is NIA Jaipur, and another is Gujarat Ayurveda University in Gujarat, Jamnagar. And likewise, in the homeopathy, there is the NIH homeopathy, and also other system, they have an intermediary system. Nowadays, almost 100, now it is a 63 peripheral center. Now it is increased about 100 peripheral center throughout the India. We are working on this pharmacovigilance system. Next. So we are giving more emphasis on the reporting of ADR/AD. That is, we can report through the pharmacovigilance center, peripheral pharmacovigilance center, through intermediary center, and that to the national coordinating center. And also there is a chances and opportunity through the ayushsurakha.com. You can you can digitally make a report. Next slide. Digital reporting system also is available. After there is a standard format for the ADR/AD, and after filling this, you can submit through digitally also. That is a good thing. I think anybody, any physician can do. But important thing is of this drug safety, it is not directly related, but it is really challenging for the Ayurvedic physicians, especially the government sectors, misleading advertisements. So we are working on this line also. Like that, so many people, so the learned people also taking medicines through getting this advertisement from the newspaper and the television also. Magic effect, or sometimes they are claiming no side effect, cure within 2 days, diabetes will be cured, cancer will be cured, like that. This is also a part of reporting, and we are taking the legal action of this. If there is any misleading advertisement. Next. Another example of the misleading is that they're claiming so many things. We are also aware, and we are trying to aware the people also not to get these medicines through this advertisement or online. So main structure is that the beneficiaries or the patient or community, they will report to the peripheral pharmacovigilance systems. Peripheral pharmacovigilance system, they will report to the intermediary pharmacovigilance system, and ultimately it goes to the National Pharmacovigilance Coordinating Center. A few examples I'm giving my experience, like that one patient was having the vitiligo, and some product they're claiming this, they'll cure this vitiligo. And after giving this, there is a huge blister, and followed by, there is a, you can see these pictures. Like that, this kind of things we have to report. It is happening in our practice also. Next. Sometimes also medical error also is happening in frequently. Sometimes you are eating some medicines, and the shopkeepers, or they're giving another medicines. So there is a drug allergy or the skin irritation of this particular medicines also very much common. Next. In my experience in 2008, when I was in Calcutta, because there is a pharmacovigilance centers, we have got a lot of adverse drug reactions, and also reported, like Zandopa, one reputed company is giving for Parkinsonism, also having that nausea or vomiting of this particular medicine. Also, we have talked with the companies, and we have resolved these problems also, like Leucoskin, like common herbal products. And so painkiller, like one medicines very popularly in the Calcutta market, that is also having the hyperacidity, had one complaint of the so many patients, like that. Even in the local applications also having some adverse drug reaction. Next. These are the common medicines which are using in the practice, also having some adverse drug reaction. it should be reported. At least we should aware about this adverse drug reaction before using it. Next. So some single herbs also having some adverse drug reaction, like Josti Modu, like Glyceria gabra. If you take the higher dose or not maintain the dose, and also some patients also getting this after the decoction, having that vomiting tendency or nausea of this particular products. Some therapy also we are giving some leaves, tender leaves, and we're using for that pain management, also having some adverse drug reaction. Next. So in a nutshell, you can say the particular one peripheral pharmacovigilance centers that reported. You see that in 2019 and 20, there is a 270 misleading advertisement in only on the newspapers, and we have documented 70 ADRs. And 2020-21 also 257 and 17 ADRs. So it is not that Ayurvedic medicines means it is using natural products having safe. Definitely there is some maybe some adverse reaction. It should become in notice. Next. So in a nutshell, we can say that there is a widespread common belief that is Ayurvedic medicine natural, is a safe, but it is not like that. Even in the Charak Sangitas, they have mentioned, in the other Sangitas, other Tantra, they have mentioned, we are dealing with some toxic, which is plant or plant material we are using, which inherent toxic. So we need that detoxification process through traditional methods. And that is the main things that we have to have more and more data of safety data of this particular system we're using. And I think if you go to the ayushsuraksha.com, you'll get a lot of informations. And also we can do report of the ADR AD through this portal. And definitely it is a good approach, I think, by the ministry. So in the end, I wish everybody Sukhayu and Hitayu and safe medication for all. This is our slogan. Thank you very much.
Thank you, Dr. Mitro. And I think that was an excellent talk on the world of pharmacovigilance when it comes to Ayurvedic medicine. And I think a very important message is clear that the government of India is putting in a lot of effort for not only the allopathic pharmacovigilance, but also the Ayurvedic pharmacovigilance. The challenges were very well pointed out with respect to the absence of journals which can publish these, and as well as the lack of awareness of where to report. But I think a long way has come where there were situations where we knew that that Ayurvedic medicines have no ADR, from being no ADR to actually reporting that there are ADRs. So definitely this is a great step taken by the government of India. Thank you for highlighting how the reporting happens through for the Ayurvedic ADRs or the ADRs due to Ayurvedic medicines. I will now take a couple of questions which are there in the chat box. So I think one of the questions which Dr. Mitro people are asking is, after reporting of the areas, what happens to the areas? Do they go for case processing? What is the fate of these areas after reporting? If you could explain that, please.
Yes, after getting this ADR reporting, then the causality assessment will be done by the local peripheral pharmacovigilance systems. They have a multidisciplinary approach, like that in Calcutta. There is a center with the people from pharmaceutical sciences, people from the microbiology, chemistry, and the modern medicines and Ayurvedic medicines. There is a multidisciplinary biochemistry people is there. So after getting this, causality will be done by this peripheral pharmacovigilance center, and they will report to the intermediary center. Then intermediary center, they report to the National Coordinating Pharmacovigilance Coordinating Center, and which is now in the All India Institute of Ayurveda, New Delhi, Sarita Bihar. So they are doing final the causality assessment and case processing, and they are reporting to this ministry for if need any action. And so many drugs already taken action by this case processing by the Ministry of Ayush. And Ministry of Ayush, they are instructing to that the licensing authority of the state government. By this, so many formulation we have taken care by this system of processing.
Thank you, Dr. Mitro, for that very clear answer. I think you have also answered the next question, which Dr. Rishikesh Kulkarni had asked about that causality, post-causality assessment. What are the measures taken for updation of data? As you mentioned, that the Ministry of Ayush is playing a very active role and is instructing the Central Drug Regulating Authority for updation of the information for the drug use as far as Ayurveda is concerned. I think we will have to end this session because we have very less time now. There was a direct question which was to me is that why doesn't allopathy take curcumin to the next level? Wouldn't that be a blockbuster anti-inflammatory drug? So let me tell the audience friend who has asked me this question that yes, curcumin has been used as an anti-inflammatory drug in many of the painkillers, and honestly speaking, I don't think it has become a blockbuster anti-inflammatory drug. So it is being used by some of the companies in combination of and also it is being recommended as an anti-inflammatory. But frankly, I think we need head-on comparative clinical trials to prove that they are better than the allopathic medicines. So with this, we will end this very interesting session on safety studies in Ayurveda medicine and hand it over back to Dr. Shambo. Thank you very much.
Thank you, Dr. Achinto Mitro, for this wonderful deliberation. And we are really thankful to both of our chairpersons, Dr. Subhrojyoti Bhowmick and Dr. Sandeep Mukhopadhyay, for being with us. And now we have to move towards the next session. And for that, we have two eminent pharmacologists, Dr. Professor Abhijit Hajra sir with us. He is the Professor of Pharmacology from IPGMER and SSKM Hospital. Welcome you, sir. And also we have Professor Trupti Swain, madam. So madam is the Professor and Head of Pharmacology at SCB Medical College, Cuttack, Odisha. Welcome you, madam. Now over to our both eminent chairpersons for the proceedings of the next session.
Professor Hajra, are you here? Professor Hajra is here?
Yeah, I'm there.
Okay.
Yeah, I'm there, ma'am, but I'm having some connectivity issues. So I'll keep my video off once.
Okay.
So
fine.
I think we are already running behind schedule. So
Right.
we'll request Dr. Parikshit to start his presentation and then we'll summarize at the end. Is that okay with madam?
Yes.
With you?
So some few
Okay,
introduction,
so
if possible, 2 lines about the speaker, Professor Hajra.
okay, Dr. Parikshit is an Ayurvedic physician with a lot of interest in product development, and as he has a good academic record. And let's see what he has to say, share with us today. So Parikshit, you can start. So I'll keep my video off. Parikshit, we cannot hear you. Yeah, you're audible, but your voice is not very clear.
Thank you, the organizers for giving the opportunity on this auspicious occasion. I'll just share the screen. So today is the screen visible?
Yes, it's visible. Screen.
Today is 12th August, and we are present here on the auspicious occasion of Maharshi Charak Jayanti Mahotsav. And also, I mention you that today is also World Sanskrit Day. So we are celebrating 2 days together. And so I'll start with a sloka from Charaka Samhita that which describes Ayurveda. And also, since it is Sanskrit Day, it will be very good to know about the basics of Ayurveda. So the sloka is Hita Hitam Sukham Dukkham Manustasya Hita Hitam Mananta Tasya Jastastam Ayurveda Sauchate, which means that Ayurveda deals with the happy and unhappy life and explains what is appropriate and what is inappropriate. Also, it deals with the nature and quality of the life. So, if we come, the basics of Ayurveda is to promote health, and another definition of health as given by Maharshi Charak, also known as the father of surgery, that Samodosha Samarnishta Samadhatu Nagapriya Prasanna Atmendriya Mana Swasthaitya Vidyate. So basically what in Ayurvedic concept we say that the balance of the bodily humor, that is doshas, healthy Agni, and good state of other muscles and tissues and their metabolic end products lead to a balanced state of senses, mind and spirit. The definition of WHO regarding health, that health is the state of complete physical, mental and social well-being, but Ayurveda adds another aspect, that is the spiritual well-being. Now I have been given a big task to find out the gaps in research in Ayurveda. So as we know, we are basically Ayurveda is based on philosophy, and all of it based on the 5 elements, Pancha Mahamutta. So Ayurveda have doors and windows. Every gap is open for research opportunities, and gaps are there. So few of the points I need to discuss regarding the research methodology in ancient Ayurveda. Right, in Ayurveda, the terms as mentioned in Charaka and Sushruta, like Kraman, which is the review of literature or means of knowledge, Prameya, Samsaya, Prayojana, Drishtanta, Siddhanta, Avaya, Tarka, Nirnaya, Vada. Tarka is the criticism. So you can see from the term that Ayurveda already has discussed research thousands of years ago. And when talking about Maharshi Charaka, he is one of the eminent scientists of Ayurveda, because all these concepts and specific clinical research or observations through which they have found that which medicines or which formulations are used in different diseases. So this is also a research. Now, what is the need for the research in Ayurveda? Because as we know, the scientific research does not necessarily mean the conventional allopathic model of research, because Ayurveda concepts are different, and when it's different, we cannot put in the same criteria as of the present modern research methodology. So strength of Ayurveda lies in the strength of its unique principles and approaches. As we know, researchers have invested entire effort to drug development, ignoring the science of Ayurveda. Researchers carried out have yielded many useful outcomes, benefited medical practitioners, and also added new knowledge. But in present time, we see there are some lacunas, because new knowledge are not added frequently, and there are lots of gaps in this area. Because even the drug research has frequently ended with negative or ambiguous results. And the important cause of such failure studies is the use of inappropriate research methodology. This is the most important factor in research or research with Ayurvedic sciences. And as we very well know, there is a methodology crisis in Ayurvedic research. Because there is a lot of research has been conducted at different levels, adopting different guidelines, methods, protocols, but ending up with research outcomes with low and poor translational value. As we see, there are many guidelines like GCP for active drugs, ICMR guidelines for biomedical research, GCP guidelines published by CDSPO, research in Ayurveda sector, or let us say Ayurveda. But what about the success and failure? As the Central Council for Research in Ayurveda Sciences, they have categorized Ayurvedic research in 4 categories, like literary research, conceptual research, clinical research, and drug research. And the only component from our aspect which we can say we have been successful is the literary research and literature development research regarding that. So, what is the purpose of research? Purpose of the research is to disseminate the results to the masses, have a database, generate data. People should know what the pros and cons of the research. So, I will start from the back process, right? Every research, the report of the publication is the final stage. So, I will start from that thing. That publication, which is published or perish. Because then they say, if you don't publish, you will perish. So, in the standard publication in research in Ayurveda is not there. There are not good peer-reviewed journals. So, these are the gaps. Journals outside the domain of Ayurveda, they have different parameters to assess the Ayurvedic research. As I have already discussed, my previous speaker, Dr. Mishra, has already told that the basic principles of Ayurveda are different from the conventional systems of medicine. So there should be a different parameters, and there is no gold standard, or the gold standard which is followed in the present day conventional research will not fit for the researchers in Ayurveda. Also, there is lack of exposure to publish in good journals, because with the Ayurvedic fraternity or the students of Ayurveda or the researchers in Ayurveda, they are not well versed in the standards of medical writing. They don't know how to write a good paper to get it published in a good peer-reviewed journal or PubMed Index journal having impact factor. So I have already discussed that there is a lack of training in medical writing. Sometimes the journals don't accept the papers because they think the publications are false and the results are exaggerated, and it is a true thing. It is false, but it is a very true thing that the results are exaggerated sometimes. This is a very much malpractice. Another thing is that improper application of biostatistical analysis. This is a very important thing I should discuss, because I have been in the academia and I have seen many theses from different universities, top-notch Ayurvedic universities from India, and many theses from on the postgraduate level, PhD level. There is a basic sense of missing of biostatistical knowledge among the Ayurvedic scientists. And simple thing, if your results are wrong, if your analysis is wrong, then your result is wrong, and then the whole project, whole research or any kind of research is wrong. So next point I will focus that on the basic principles. As we know that the basic principles of Ayurveda comprises of the 5 elements, that is what we term Pancha Mahotsav. That is the space element, air element, fire element, water element, and earth element. And based on these and these permutation and combination, they produce the Tridosha theory. The Tridosha are the 3 basic body principles based on which a body is a state of normalcy, or when they are in deranged state, the body is in a state of imbalance or the disease state. Then there are other factors, what we call dhatu. Dhatu is the tissue system, or what is the bone, all these things, and the excretory system. These all comes under the basic body system, the basic principles based on which a body is formed. And also the important concept of Agni, because it is a fact that it is also believed that due to the digestive fire, Agni, we can roughly correlate with the digestive fire or the fire element of the body, due to which many of the diseases manifest if it is deranged. So the concept of Agni is there already. Many of you may be knowing the concept of Prakriti. It is a very important thing, like in Ayurveda, it's a genomic level concept. So, based on Prakriti, a person's Prakriti, the drug is provided, depending on the property, it is that the drug, how the drug acts on the body. And also a new concept has been developed by Professor M.S. Bhagiyathan, that is the concept of Ayurveda biology. So all these present day parameters in research need to be understood on the basis of Ayurvedic principles. That only if when these principles are understood properly and when they are understood on the basis of the present day parameters, then only the research, each of the research gap of Ayurveda will be diminished. Now coming to the point of drug research. Already Dr. Miklo has focused on the different aspects of drug research, adverse reactions and other things. So one of the important feature is the lack of funding. Also non-participation of industry, means Ayurvedic industry in the drug research. Because there are not much funding and government funding is limited with limited resources. Another thing is that new proprietary medicine, every drug manufacturing company is producing some of the formulations, but those formulations prepared by them, which are not classical formulations, does not undergo proper trial. Not in toxicity studies, not animal studies, nor safety studies, and also proper clinical trials are also not conducted properly with proper research methodology. There are also safety issues and also the issues with quality control. Monitoring boards are there, drug control boards are there in every state, but they are not strictly monitoring any GMP certified companies, manufacturing units and everything. And also lack of stringent monitoring are also there. Another important factor is also been discussed in the terms of pharmacovigilance was the Magic Remedies Act and the false claims. Also in Ayurveda research, there is not much focus on insurance and compensation to the patient. If something happens, everybody is thinking, see, the Ayurvedic drugs or formulations are extremely safe, they are devoid of side effects. Even the researchers also think. So during the clinical trials, they are not keeping in mind regarding the insurance and compensation. Now, after the NDCT rules of 2019 has come up, all these things are specified that this should be keeping in mind. But till now, researchers which has been undergoing on Ayurvedic formulations, this kind of topics or facts are not being kept in mind. Now, another concept, and it is one of the present things Ayurvedic research is based on, that is the bedside to bench approach or the reverse pharmacology approach. As we know, for centuries, we are using Ayurvedic medicine and effects are noted. Sometimes the adverse reactions are also noted. It is not that there were no adverse reactions. Otherwise, in our classical text also, there are indications and contraindications for every medicine. So, from that ancient times, the times of Charaka, Susruta and Bhagavata, they have noted adverse reactions of the drug. So, those scientists have noted adverse reactions and they have also noted what to be kept, what are the most serious adverse effects and this kind of thing. I'll be going on fast because I think the time is very short on our hands. So another important aspect, everybody thinks about the use of heavy metals in Ayurveda. If you be knowing, around 15-20 years ago, there was an article came up in JAMA, Journal of American Medical Association, that getting Ayurvedic medicines contain heavy metals. Yes, it contains heavy metals. We all know Ayurvedic medicine contains heavy metals because Ayurvedic medicine is not only all about herbs, it is also about metals. And be surprised that in Ayurveda we use one of the most toxic metals as medicine, that is mercury. So in Ayurveda for centuries we are using mercury, but not mercury in the normal form. There are around 18 procedures of purification of mercury to which the mercury is prepared and detoxified, and then it is used as a medicine with other metals, other herbs. And this metallurgy or the art of using metals with minerals and with herbs in Ayurveda, there is a stream called Rasha Shastra. So, giving one example I can share that during COVID time, everybody in India was advised by Ministry of Ayush to use herbal medicine, some of the immunomodulators which have been used, and the kitchen, because in Ayurveda there is a concept, kitchen dispensers. Because many of the medicines used in Ayurveda, we will get readily available in our kitchen only. But nobody from Ayurveda, shocking for me, that nobody from Ayurveda discussed about the super effects of the Rashoshadi, that is the metal in effects of COVID-19 patients or due to the immunity of patients or normal people or having having immunomodulator effect. Like one of the important Rashoshadi we use in our daily practice as a physician, that is Mahotsav. Its most important composition is Paras and Gandhak, that is mercury and sulfur. So while talking with Rasasastra, it will take many days. So there is a scope for discussion, future discussions with Ayurvedic metals and minerals in different approaches for different diseases, mainly on the immunomodulatory effect. Now we come to the aspect of the personalization. As already everybody knows, Ayurveda is unique with its concept of one model doesn't fit all. Because in modern medicine, we are giving a particular molecule to all the patients. But in Ayurveda, there are thousands, hundreds, and lakhs of formulations. And sometimes what happens, one formulation I give it for a particular disease doesn't fit the person. Because while treating the patient, we have to look for different basic principles of parameters inside the body. One of them is the quantity, Agni. And this kind of things is based on this, we choose the medicine and we give the patient. So personalized medicine and researches on personalized medicine, there is a large gap. And now another thing is that it is very difficult to establish a research model. Because suppose if I take a formulation, Ayurvedic formulation, and I give it to 100 patients with different toxicity types. But if a same toxicity type is given, it will act more or less in the same pattern. But if we give it 100 different patients with different toxicities, the medicine will not act in the same pattern. So sometimes you will see exaggerated effect, or sometimes you will see diminished effect. So when planning a research model, it is very essential to the genomic type effect. So research models need to be specific according to the study, according to the medicine also. Now, another concept in Ayurveda which is very much triumphant present days is Ayurgenomics and Ayurgenomics. As I was discussing, Prakriti, the body is made of the Dosha theory, that is the Vayu, Pitta and Kapha, based on which the disease factors are there, or the health factor is there. So, Government of India already had a research project on translational research involving genomics, and they have seen that particular genes and compared with our different varieties. Now, another concept that, as I already discussed, personalized medicine, there is a concept of personalized nutrition. So, here is a scope and gap of research that, as I already said, different food also. If food is a medicine, then food also acts differently on different quantity types. So nutrigenomics can be a new field, Ayurvedic nutrigenomics, and already there is a coined term, Ayurvedic nutrigenomics, that is just go to Google it. So ionucleogenomics can be another area of expertise of coming future. A lot of researches can be done and the gap can be filled with ionucleogenomics approach. I have already put up a book chapter based on the ionucleogenomics concept. It was published in a book named Functional Foods and Nutraceuticals. Also, there's publication that is inspired into evidence. And another concept which is being discussed adverse reactions, which Dr. Bishu has already discussed. The government of India is working. They have all different kind of pharmacovigilance centers and everything. But one thing, there is no data. Everyone is concentrating on pharmacovigilance, on the adverse drug reaction. What about drug reaction? What about the effect of the drug in the whole population? There is no data. And I think worldwide in many universities, there are pharmacoepidemiology. One of the top universities like Harvard, they have a separate department full of pharmacoepidemiology. So they are working on the drug effects in the society, drug effects in the population. So right that I have conceptualized that why cannot be a IU pharmacoepidemiology. It's department or a system dedicated on the drug use, because we are using drugs for many days, many decades, many centuries, but there is no database, there is no data, and the basic research data are also not there. Then what I can say, we need to develop to do any research on Ayurveda. Everybody is talking about GCP in Ayush, clinical trials in Ayush, but the basic first step in research should be the research in Ayush pharmacoepidemiology. Then after we go to proper clinical trials and everything. Right? What the Ayush pharmacoepidemiology is the methods of epidemiology in the content area of Ayurveda and clinical pharmacology. There is a definition, you can already go through that. There are some publications, and based on that, I have some basic level researches I have done, conducted. Right? In Ayurveda, not only in Ayurveda, present terms, there is a emphasis on importance of the spiritual and the mind effect of in clinical trial on medicine having effect on the mind and the spiritual effect in the spirituality. So one of the parameters is quality of life assessment. If you undergo database search, there is no research related to quality of life assessment. It is a very vast area and it comes under the pharmacoepidemiology approach. So I have done research, some few works on that, and also already many works are there. There is an ample scope of doing research because we need to establish a database on Ayurvedic drugs used in the among the population. As I discussed, we are using heavy metals in Ayurvedic drugs, but there should be a database that which particular medicine is taken by how many people on also on which particular diseases it has been prescribed. So there is a scope of improving the database in regarding research in Ayurveda. Then the most important facet is the clinical trials in Ayurveda. As we know that government of India have already put up the Ayush Ministry, and they have started GPP in Ayush, GMP in Ayush, GPLP in Ayush, good agricultural practice in Ayush. These should be again not confined to the scientific community, but these all things should be given or taught from the undergraduate level to postgraduate levels, and practical oriented training programs should be organized. And the important areas of clinical trials and research in Ayurveda, like one of the most important thing is that the therapeutic, Panchakarma therapeutic, like already said, the present day modern science we are using grinding effect and masking effect, all this stuff. But in Ayurveda, sometimes it is not possible to follow that gold standard of grinding and everything because procedure. In Ayurveda, there are suppose Panchakarma, there are different five, six, seven or many procedures of therapy. So every therapy procedure is different. So if you are comparing two different therapies, you cannot grind the therapy. So different what I say, research methodology should be developed, and it is in the process because the Ministry of Oil and Central Council for Research in Ayurvedic Sciences, they are working days and out on this, days and night on this thing, developing new protocols, but needs to be, those protocols need to be standardized. Then another important part is the use of metals in Ayurveda, and that is a very important area to explore. And another is the Rasayana or the immunomodulators, because in present day recently we have came up with a deadly disease or deadly disease COVID-19, which has impacted our life globally. So there are scope with Ayurvedic immunomodulators. So we, as a preventive measure, as a promotive measure, we can in future there are more different kind of viruses will be there, new diseases will come up. So this is a way we can go out for promotive and preventive measures. But another only thing is that extensive researches should be done, and not only involving only Ayurvedic scientists, because as we all know, research is a collaborative process, and multidimensional, multidisciplinary teams need to be involved in it. And as I just discussed, the basic research is like subjective well-being research, research on subjective well-being, research on quality of life. Another thing is in Ayurveda, I have seen there are many parameters, Ayurvedic parameters, and there are many subjective parameters that well-being, like many questionnaires are also there. But those questionnaires are not standardized. Like for example, WHO has a quality of life questionnaire, WHO POL, So the questionnaire is standardized based on different geographical locations, and also the ethnicity of people, and also the languages of the people. Like that only, the Ayurvedic questionnaire should be standardized, which I have seen is missing. There are no standardized questionnaires for different parameters testing of diseases in Ayurveda. Another concept, what we have many of people have already seen, have undergone, because in India people take many kinds of medicines together, like conventional allopathic medicine, and around that, with that they are taking herbal medicines, or what we can say phytochemicals, phytomedicines, also you can say Ayurvedic formulation. So this is one of the good, the integrated approach to treatment can be done, and researches have already undergone. There are many researches have undergone with the Ayurvedic adjunct therapy. One of the researches was performed by Professor P.K. Devnath. Many of you pharmacologists may be knowing Professor P.K. Devnath was an eminent Ayurvedic scientist. So he had done during his time many adjunct therapies with Ayurvedic medicine. One of his researches was on pulmonary tuberculosis, where with the conventional anti-tubercular treatment, newly diagnosed tuberculosis patients, they have added adjunct therapies with Ayurvedic formulations, single herb or multi-herb formulations. Another study was done with diabetes, in diabetes foot ulcers. Another therapy was done in chromium supplement, albumin supplement type 2 diabetes. So there are many trials have gone, but more focus and more research target oriented studies need to be done. So in the end, what we can say, research in Ayurveda is a collaborative process. People who are interested in research, who are doing research in Ayurveda, researchers in Ayurveda, they should have the proper knowledge of the subject, proper knowledge of the education system needs to incorporate all those important aspects into the education scheme. Regular updation with the training program needs to be done. Another important aspect is empowerment and not last collaboration to be done with different fields. So we can say that validation of Ayurvedic principles through enzymology, immunology, and bioprocess can be done. These are the new areas. New immune diagnostic tools for Ayurvedic diagnosis can be done. Understanding the pathogenesis, disease, also the drugs used and how the drug is working on the body, that can be done. Potential transgenic plant and with concentrated active molecules. This kind of research is going on. The mechanism of metals and minerals preparation on isolation and activation of organometallic molecules, like already incinerated gold bhasmas are used as nanoparticles in Ayurveda. It's already being used. Standardization and with cost-effective procedure drug development and pharmaceutical preparation. Innovative knowledge with intensive products and services. Biotechnology-backed screening tools for new drug development, and many other things to go. We also need to utilize the strength and opportunity, minimize the gap on the staff content. Vision should have science is the power of knowledge and experiences is the mother of science, as we know. Every observation provides knowledge is got to be utilized for betterment, and approach documents have to be prepared, follow hand, and knowledge on change of the lacunas, and these gaps, the more the gaps should be bridged, the more the researches in Ayurvedic sciences will be advanced and more scientifically evolved. So with this, I thank you the organization for giving me the opportunity to come and speak in front of you. Thank you.
Hello.
Yes. So Parikshit has given a very broad overview. So Tripti Madam, would you like to summarize for us?
Yes. Okay. Professor has okay, you are my co-chair. I am happy to have you, and Dr. Shambo, Dr. Subhrojyoti, senior pharmacologist of West Bengal and Ayurvedic scientist. I know many scientists are here. So I really appreciate and congratulate ISCR, East Zone and IPS West Bengal for the initiative of celebrating Maharshi Charak Jayanti, and I really feel privileged to be part of this celebration also. While hearing from Dr. Devnath, I fondly remember Professor Devnath of IPS West Bengal, who used to come to Odisha and who was facilitating the development of homeopathic science in our state, Odisha also. Well, we know that Ayurveda is one of the oldest science and has approached the health in a real holistic and personalized way. Though there are certain limitations, but here is the science which always says that there is a personalized approach. The science, this is a science of 1,000 years and it is real pride of the Indian indigenous system. So this thousands medical concept, which is already imbibed in our system through homeopathy, Ayurvedic and also has ability to treat many chronic conditions, as we all know, like diabetes, arthritis, asthma, which is not, we have not come to the concluding statement so far as modern medicine is concerned. So that way it is a very, very important concept. However, as highlighted by the speaker, there is a lack of scientific validation in different various concepts. And this precious gift from our ancestors is probably not getting the real importance that we need to give to the science. There is a clear and urgent need to fill the gap in the knowledge for which this topic is saying itself. And we all have to have some effort to make the science more prominent. Coming from bedside to brains, because it is already there in the system, as highlighted by the speaker. So reverse pharmacology approach. So we all need to put the effort to make it a science from bedside to the bench and in a proper way. So again, I was just going through, we heard about methodology crisis in the science. So that is really, I hope it's a problem as I was going through the literature. So well-adapted research methodology for this science is also the need of the hour. Then our presenter also focused on the funding thing, but I think Government of India is taking adequate step to generate funding and all. I don't think there is a real problem. So we all agree that there is a need for improvement in the methodology system and all. At the last, I would always I feel happy that the attempt of West Bengal Society of Pharmacology will certainly encourage the young researchers in this field to work in the various area for the research and development and promotion of the science in our country. Thank you.
So thank you, Madam, for giving our views. Now, Purikit, you have spoken about many gaps and challenges. Now, in the chat box, there's a question that what measures would you suggest for filling the gaps? Now, if you look at it from the point of view of average Ayurvedic practitioner, a practitioner in the field who is aiming to use the principles of his sciences to give a holistic healing approach to his patients. Now, of course, there are some remedies which are available, some areas where maybe potential remedies are available, and there are many areas in which no remedies are available. That happens with every system of medicine. So given the many challenges that you have mentioned, can you identify the top 3 pressing issues to your mind for both for Ayurvedic practice and research that needs to be addressed from all the stakeholders in the field?
Starting with what I started from saying, research methodology should be according to the basic Ayurvedic principles, because what we have seen that the gold standard of research is that we are doing grinding. But in Ayurveda, you cannot do that because going for the therapeutics and everything, because the smells are the different medicines, they have different smells, they have different tastes.
Okay, so this is one issue where Professor Urmila Thatte also mentioned a little while back that we cannot do a cut and paste approach for the methods of modern medicine. We cannot perhaps directly apply to Ayurvedic remedies. We may have to innovate and think about novel ways. So that's one. What else?
Now the database, because everybody is talking about Ayurvedic medicine is safe, and Government of India initiated on adverse reaction. Pharmacovigilant centers they have started, but what I feel database is needed on the use of Ayurvedic medicine in the population. In that aspect, I have started, or I have said that pharmacoepidemiology approach should be needed, and pharmacoepidemiology centers should be started, because that is application for both modern science, modern medical science.
Okay, your point taken that pharmacoepidemiologic approaches to Ayurvedic medicines and drugs are needed. That's a difficult area actually. Applying pharmacoepidemiological concepts to build up databases, not only about how much drugs are effective in addressing questions of public health, but also the safety aspect. So that's 2. And if I may ask you to give us a third most pressing issue.
Third is that the knowledge regarding the Ayurvedic science. Ayurvedic scientists, while as per my experience, when we are in undergraduate level of PG courses, in-depth research statistics were not taught to us, and aspects of clinical trials, how to conduct clinical trials properly, these were not taught. So education is one of the important things, and regularly updating the knowledge and training is one of the important aspects. Also, Ayurvedic physicians or scientists, they are not inclined to do collaborative research, but research cannot be done alone. Multidisciplinary collaborations are needed to standardize anything or do any research on anything. That will be my third.
Okay, and this is a point which is after my very heart. See, what I personally feel is that we may be practicing one system of medicine. We may be so-called Western medicine or modern medicine practitioners. We may be Ayurvedic practitioners. We may be homeopathy practitioners. We may be practicing Tibetan systems of medicine. So each system has its own principles, own views of how to diagnose and tackle individual problems. But when it comes to research, this sort of compartmentalization perhaps should not exist. Okay. Ultimately, what is the goal of research? To generate new knowledge and to refine existing knowledge so that we can arrive at solutions for problems for humankind, problems for animals and all that. So whatever be the source of the knowledge, it can be a plant, it can be a chemical, it can be a mixture. So there should not be any compartmentalization in research, and real research will require teamwork, will require multidisciplinary input. So building that sort of climate, I think, is a very important issue in India and quite challenging at the moment because of the way we are accustomed to thinking. So thank you, Parikshit. You have highlighted wonderfully many known aspects, known challenges, as well as new areas like pharmacogenomics applications in Ayurveda, pharmacoeconomics applications, pharmacoepidemiologic applications. So thank you. And I'll now hand over the microphone back to Shambo to sum up this excellent proceedings for the day. Shambo, over to you.
Thank you, sir. Thank you very much. So we are really thankful to Professor Abhijit Hajra sir and Professor Trupti Swain madam for agreeing to be the chair of this session. And we are really delighted to have this wonderful deliberation from Dr. Parikshit Devnath. So we all are actually a big fan of his father, Devnath sir. So we are really remembering him today. So may I now request Dr. Subhrojyoti Bhowmick, who is the chair of ISCR East Chapter, to say some important notes on today's program.
Thank you, Shambo. And at the outset, let me thank all the illustrious pharmacologists who are present here. I have my own thesis guide, Professor Abhijit Hajra sir here. Thank you, sir, for taking out time and being here. Professor Trupti Swan, she is one of the leading pharmacologists in Eastern India. Madam, thank you very much for your presence here. We are honored to have you. We had Dr. Urmila Thatte, madam, who is the mother of clinical pharmacology in India. We also had Professor Shantanu Tripathi, sir, who is also the leading clinical pharmacologist in the country. And we are really honored to also have Dr. Sandeep Mukhopadhyay, who is at ICMR NICET. I would say this was an our great speakers, starting from Dr. Parikshit Devnath to Dr. Mitro, to Dr. Shoiban Mukherjee. All of them have focused that no matter the branches of medicine, whether they are Ayurveda or it is homeopathy or even allopathic medicine, the focus of patient safety and patient care is of ultimate importance. And we at East Chapter of ISCR would really like to focus this over a period of time. I would again thank Dr. Shambo Samajdar, who has put together this entire program. And I think you will see many more programs in coming days where we will involve not only the allopathic physicians, but also the Ayush physicians in the region to have a holistic care so that we can have a holistic patient care in this part of the country. Again, thank you very much, one and all, for being part of this important initiative. I wish you my gratitude as well as my greetings for the upcoming Independence Day. My greetings for the happy Independence Day ahead. Thank you very much. Jai Hind.
Thank you. Thank you, Dr. Bhowmick.
Thank you, Dr. Bhowmick. So at the onset, now I am really thankful to Innovocare E-Academy also for actually having us or for providing us the platform, this online platform. So thank you, Innovocare E-Academy. So we are really thankful to all the speakers and chairpersons. So we have another program in the upcoming days. Hopefully we will meet you soon. So with that, thank you all. And again, advanced Happy Independence Day. Jai Hind to all. Namaskar.
Thank you. Thank you. Dr.
Thank you.
Shambo.
Bolo.